Welcome to this session, which is billed as a quickfire session. What we're going to try and do is run through three brief 10-minute presentations, and then if you have any points that you want to bring up, speakers are happy to be interrupted, but if you can keep the majority of questions, we'll have a roundtable session at the end. I've got on my left Peter Urwin from Leeds University, David Cooke from Hutton Institute, and on the right Mark Taylor from Hutton as well. And obviously we're concentrating in this session on novel breeding techniques for some of the issues that we have in potato production. So we're going to start off with Peter first.
Okay, thank you very much. If I stand here, does this microphone work? It does, it does. So I have 10 minutes just to briefly explain to you one of the projects that's going on in our laboratory. The group is at the University of Leeds, and the project that I've got 10 minutes to quickly describe to you is one that's entitled New Potato Varieties with Resistance to Late Blight Potato Cyst Nematode with Reduced Bruising and Improved Processing Quality. To tell you who's involved with the project is led by the Sainsbury's Labs at Norwich by Jonathan Jones. Jonathan leads the project, and he's down in Norwich. And Jonathan works on natural resistance of blight. So he's concentrating on that aspect of the project as well as looking at it overall. Myself and Howard Atkinson are at the University of Leeds, and our research is focused on plant parasitic nematodes. And so in this project, we're bringing our expertise in biotechnology to control potato cyst nematode, and we're donating those genes into the project. There are two companies involved, BioPotatoes, which are a very new company that have been formed, and they have the specific objective of catalyzing the development of GM potato varieties. While they're a very, very small company and have only been in existence for 3 or 4 years, Simplot is the US leading potato processor, and it has a very strong commitment to biotechnology. In fact, 2 of its GM varieties are in the final stages with the FDA to gain approval so they can be grown commercially from 2015. And they're potato varieties that are anti-bruising. So they're donating the bruising aspects that are in these new things. The project, as well as being supported by these 2 companies, is also supported by the AHDB. So the objectives of the project, to create some DNA that has late blight resistance and to transform the 2 varieties that the companies have asked us to focus on, Maris piper and Agria. So that will be 3 resistance genes, 3 natural resistance genes that will give late blight resistance into Maris piper and Agria. The second objective is to validate the efficacy of DNA, 3 genes, that will generate low acrylamide in the potatoes and bruising control. So the genes that we're using for low acrylamide are a couple of enzymes that together act together to lead to the formation of acrylamide in potato. If you silence these genes, if you knock out the function of these genes, then they can't work and the acrylamide can't be formed in the potato. Similarly, for bruising control, there's an enzyme that leads to bruising, and if you silence that gene using GM technology, that gene is silenced, it doesn't have a function, and you don't get bruising. Knocking out the gene has no other effect on the plant. So the plant is robust as any other plant, but you don't get the bruising phenotype on the tubers. The third objective is the one that I'm going to spend one more slide talking about after this, and that is to demonstrate the efficacy of GM resistance against PCN. Potato cyst nematode, very important problem, as you all know. And we have two types of technology that we've put together. We've got one type of technology that disrupts chemoattraction. The nematodes in the soil have to detect the plant and they have to get to the plant. And we can make a GM potato that disrupts that chemoattraction. Put very crudely, the juvenile animals can't smell the potato plants and they can't find their way to the potato plants. And also we have some technology that if the nematode does get to the plant, it stops it feeding. So we have two technologies for PCN resistance. The fourth and fifth objectives basically bring all of these genes together in a stepwise manner to act together. So objective 4 brings late blight together with the low acrylamide and bruising control. And then objective 5 brings everything together. So ultimately, we'll end up with a potato variety, a GM potato variety, that is resistant to late blight, doesn't bruise, has low acrylamide content, and is resistant to potato cyst nematode. So they're the objectives of the project. That describes everything that's going on in the project. And one of the talks that's coming, you'll hear more about late blight. What I just want to spend one slide talking about is the resistance against potato cyst nematode. You can see here a picture of potato cyst nematode on a potato root. Please bear in mind that this potato cyst nematode is only about 0.5 millimeter in diameter. It sets up a specialized feeding cell in the root that we call a syncytium. This cell would not be there if the nematode was not there. The nematode specifically induces that structure. And if I just cut through that root and take a picture, you can see the syncytium here. Sorry, I should say I've stained it blue. It's not normally blue. If you cut through that root, you get a cross-section that looks like this. You can see the phloem and the xylem that do the jobs of carrying nutrients around the roots and to the tubers. And you can see there a cross-section through the syncytium. It has a number of nuclei in it. Cells have collapsed together to form this structure. And you can see maybe, not from the back, but hopefully you can see there are 1 or 2 cell walls that are collapsing together that form this multicellular structure, which just becomes one structure because the cell's walls dissolve. And the nematode feeds from that structure for 6 to 8 weeks. The nematode feeds using an enzyme that we call a protease, so it has to digest protein, and the enzyme that digests protein inside the nematode is a proteinase, and this is what the proteinase looks like. This is the active site of that enzyme, with the key part of the active site shown in yellow. We can generate potato plants that have an inhibitor, and that inhibitor will dock. He's the part of the inhibitor that will dock into this active site, and it will stop this proteinase from working. This proteinase is made in the nematode digestive system. It gets into the plant, it sets up the syncytium, it tries to digest the contents of the syncytium using a proteinase, but if the plant is making a proteinase inhibitor, the nematode takes up the proteinase inhibitor along with everything else, and the enzyme can't work, and therefore the nematode fails in its reproductive success, and you get very few nematodes, and those nematodes produce very, very few eggs. So that's the proteinase inhibitor technology. The other technology that stops the nematodes finding the roots is host location and migration to the root involves chemoreception, i.e. the nematode, again put very crudely, has to be able to smell the plant. And we know that some small molecules are taken up by those sensory neurons. So those nerves, just to put it in context, you might understand those nerves that are in your nose that allow you to smell have openings to the environment, and certain small molecules will be taken up by those nerves and travel back along those nerves. And one of the things that does it is a very small protein that we call a peptide, and this just tells you what the sequence of the peptide is. And we can link that peptide to a dye, and we can see that those open nerve endings where the animal senses its environment takes up the peptide and it travels back down the neuron to the cell body and then forward again to the nerve ring that you could think of as the brain of the animal. and it disrupts the functioning of those nerves and stops the animal detecting the plant. So we're disrupting chemosensation. And if we just have an agar plate and we put nematodes at the edge of the agar plate and attractant in the middle, you can see that normally nematode tracks get more and more dense as it gets closer and closer to the attractant. If we soak the nematodes, first of all, in this very short protein, this peptide, they're unable, even on an agar plate, to find their way to an attractant. If we get the plants to make the peptide for us, if we generate GM plants that secrete this peptide from the roots into the soil, the juvenile nematodes can't find the plants. So this would affect the previous technology. The previous technology would only affect animals that get into the plant and have to be a parasite of the plant. This technology would affect all the nematodes in the soil. But nematodes have a way of cleaning their own neurons. So if a nematode was to come into the root structure of a potato plant excreting this peptide, it would be affected. It would probably move away from the plant because it's not attracted to the plant. It would clean itself, and therefore kind of like there would be no effect on that nematode. So it's not a lethal, it's not directly lethal to nematodes. It just stops them finding the plant. But for plant parasitic, for PCN, those animals have to get into the plant. They have to get into the plant. Until they get into the plant, they will not feed. They're running on reserves. They're running on lipid reserves. They're using their own fat. And once they're hatched, they have to get into the plant. If they don't get into the plant, they will die because they will run out of lipid. And they can survive for maybe 1 to 2 weeks in the soil as juveniles. So ultimately, this technology does kill those animals too. So they're the two technologies that we're using to control plant parasitic nematodes. Last slide on where the future of this technology might go. There's synthetic biology that allows you to put an awful lot of genes into a plant, and really that's what we're doing here. There are a number of traits, each of those traits, number of genes. You can see we can put a big lot of genes into a plant, and so that's what synthetic biology is. You will have heard in the media of something called genome editing. It has applications in the medical world as well as in the plant and agricultural world, where you can tweak a plant's genome and alter its own genes. And that's called genome editing. And in the United States, that wouldn't be classified as being GM. In Europe, however, that would be classified as being GM because it undergoes a GM process, although at the end of the day, it's not genetic. It doesn't contain any foreign DNA. So in the States, genome editing would be classified not a GM plant. In Europe, at the moment, it would. Okay, so very quickly, 10 minutes. I'm sorry if I've overrun. That's our project. Okay, thank you very much.
Thank you very much. Okay. If anybody's got a quick point, they can bring it up just while David's getting ready. But otherwise, we'll take them at the end. Andy, do you want to?
Does that peptide, is that PCN specific, or does it also affect other nematodes? No, the peptide would affect any nematode around the roots of the plant. But it would only kill those nematodes that actually would run out of reserves because they have to get into the plant.
Yeah, I always thought polyphenol oxidazes were quite important in first defense against plant pathogens. Is there a risk in making a bruise-resistant tuber you might increase susceptibility to infection?
So the polyphenol, you're quite right. They're quite right. The plant has a number of polyphenol oxidases, and their functions overlap. All I can tell you is that all of the data that has come from Simplot, who have researched that particular trait, would indicate if you knock out that particular polyphenol oxidase, then you get no bruising in the tubers, but the plants are as resistant to all the pathogens, as resistant as the parent plants. So that seems to be a polyphenol oxidase that is majority, most of its function seems to be in the tubers in bruising.
Okay, thank you for that. If you've any others, we'll take them at the end. David give us an insight into blight.
Okay, thank you very much. So my name is David Cooke and I work at the Hutton Institute up in Dundee. And my brief was to present to you in 10 minutes the next 5 to 10 years of late blight management and where I see the future going. Quite a challenge, and as Mark Twain said, don't make predictions, especially about the future. It's quite a difficult one, but what I wanted to do is take you on a whistle-stop tour through some of the current research projects which are intending to provide those tools for the next 5 to 10 years. So that's what I'll be running through. An overview really is just thinking about the pathogen triangle. We've got the plant pathologists always think about the pathogen, the host, and the environment. So we're looking at tracking changing pathogens, why we need to do that, how to predict blight infection and understand blight infection and spread. And the idea for that is obviously to give smarter spray advice based on this, precision agriculture, this new knowledge, in-depth knowledge that we heard about in the first session today. But also I wanted to touch on a longer-term strategy of improving the host. So I'll touch on the use of technologies such as CRISPR we've just heard about, and where we're going with that. And one of the threads I wanted to run through this is the idea of the amount of data we have now, and we should all be sharing this data in a sort of collective way to improve UK agriculture and integrating these into models as a kind of core activity. Lots of information here. I really intend you to read more the presentation, and I certainly don't plan to go through all of them. But it was to give you a flavour of the types of funding and the range of funding that is being used. And these are projects just the James Hutton are involved with, but funded by, of course, groups such as AHDB Potatoes, industry, Scottish Government doing some of the underpinning work, which provides that background for the applied knowledge in these other areas where we've got new tools and products to aid management. So again, AHDB funding PhD projects for the future and working with industry on new technologies. And lastly, some of the applied modelling side. So really just a flavour of the range of research that we're doing. I'll be touching on a few of those. You're familiar with this, with me talking about this. This is the sort of understanding, knowing the enemy type of work. Again, continuing to be funded through the Fight Against Blight campaign, Scouts providing us this valuable data on the pathogen populations and how they're changing over time. What you probably haven't seen is now we're integrating these results into a European database to give us that broader perspective, plotting the data in a finer resolution in space, so we can see which lineages are where. Immediately you can see, that Northern Ireland has a different population from parts of the rest of the United Kingdom. And moving on to this rather pretty but complicated novel genetics tools we're using to understand the way the pathogen's evolving. And really the main benefit there to think about is the way we can track inoculum on a European scale. So that's kind of knowing the enemy, understanding. And I see that as important future-proofing for all the other work we're doing, breeding, et cetera, et cetera. Again, I'll just touch on this one briefly. This is kind of redesigning the Smith period in a nutshell, I suppose. It's looking at improved decision support. Siobhan, who's sitting over there and has a poster outside so you can speak to her, is doing this project where we're looking at redefining the thresholds. And we're not just sort of plucking a number out of the air. We're doing this in a scientific way to really understand both the temperature. So there's some data here at 10 degrees. No time to go into details, but at 10 degrees, for example, where the Smith cutoff is, but looking at also at different humidity thresholds to define more finely and appropriate to the contemporary genotypes where we should be setting these thresholds. We then will take that, integrate that data, comparing it to the data that on the Smith period outbreaks for the last 10 years, which we've assembled through that long-term research project, to understand where Smith is failing and how we can improve it, and then validate it in the future. And that links into modelling work as well that Pete Skelsey is heavily involved with. I'll mention Pete a few more times. Moving on, that model assumes we know we have just a general pile of inoculum. We assume the inoculum is everywhere, and when the conditions are right, blight will emerge. However, this project, which is funded by Innovate UK and has multiple partners that I'm sorry I don't have really time to give credit to all of them, I mean, Burkhardt are making the traps, crop performance, managing the project, Rothamsted are critical in terms of technology. What we're looking at is trying to detect inoculum as it actually arrives in crops, using a spore sampler, methods to automatically degrade and destroy that, not to degrade the DNA, but to release the DNA from the spores, some sort of an amplification step which enables us to detect Phytophthora, linking that to a dispersal risk model so we can say, yeah, there is inoculum in your crop, but where's it come from and what does it mean in terms of risk, and translating that to messages to allow the grower to make more informed decisions about blight being present. It's all in development at the moment, but that's where we're heading with it. Other Innovate projects are also exploring other forms of in-field spore detection, but also using drones, aerial reconnaissance, as we heard about in precision agriculture. So there's a lot of work going on in this area. Again, I'm going to stress this advocating of sharing information and in-field metadata in precision agriculture. Moving on, I mentioned late blight and late blight resistance. We have to, you're not exposed to the world necessarily of the massive amounts of genomics that are going on, but the potato genome and Phytophthora infestans genomes have been sequenced, and we're learning more and more about these. A couple of images here. This is a potato genome with a lineup of a flagging of all the resistance-related genes. So there's hundreds and hundreds of them. And what we also understand now is the pathogen itself. This is a micrograph of the pathogen invading, and you can see this little sort of peak here as it penetrating a cell and feeding. We understand a lot more about how the pathogen does that as well, and all the effector genes it has. So we're looking at the interactions between the pathogen genes, the host which recognizes them, and the way the pathogen then evades that recognition. And what we're trying to do is identify durable resistance, so pathways in the pathogen that cannot be avoided, cannot be circumvented. And of course, then we want to integrate those results, those genes by either conventional crossing, GM, or gene editing technology we've just heard about. However, that's one thing, but you've got to deploy this stuff right. So efficient deployment using landscape models is something which is important in the future as well. Rushing through, another project, this is funded through the Horticultural Potatoes HAPI initiative. Again, multiple partners. Principally, modelling is done by Rothamsted Research. ADAS and SIUC are running the field trials, and we're integrating with that and passing data on and processing these samples. In a nutshell, this project is aiming to harmonise the way that fungicides and host resistance, your two main weapons against potato blight, can be used long-term in a sustainable sort of way. So there's always a risk when you introduce a new fungicide mode of action that when you use it, when you apply it, it provides a selection pressure and the pathogen can evolve in sensitivity. But we can use resistance to slow that process because you're suppressing the population. On the other side, whenever we employ a new R gene, unfortunately, we don't actually deploy many new R genes that are being used, and so that's a different matter there. But we're working on particular R genes that have been introduced, and we select for virulence, but you can use fungicides to suppress that process. So the whole, we're testing these two hypotheses in large field trials, plotting the way these epidemics move, and understanding the way we're working in the future to try to provide that durability through modelling. And my last slide comes back to modelling. And I think, again, it's stressing the fact that we have this vast amounts of data now. It's an image put together by Pete Skelsey, who is a modeler, very interested in spatial models. I share an office with him. And, for example, here we have the IACS data from the Scottish Government on all the potato plantings within Scotland. So that's, it's not just a fuzz we've sprayed on the map. That's actually the locations of all the potato fields. So one can zoom in and look at the integration and the relationship between seed and wear, disease spread between the two, to explore hypotheses by building that sort of spatial data into crop growth models, disease risk models, pathogen dispersal models, and climate change, looking to 10 years or 80 years in the future. So we're looking at, Pete's now currently running models on where blight management will be in the future. It's going to be warmer and drier, but does that mean it's going to be easier to manage? It's interestingly, some preliminary results suggest it's not always, no, because the crop season will also change. Okay, so the general consensus here, general idea is then merging all this information together, this and more data, to create this sort of virtual lab to use in a heuristic way. Just means sort of using it to test hypotheses and direct further research. And I can see I'm up to my 10 minutes, so I shall stop at that, and I'm happy to answer questions now or later. Okay, thank you.
Ready? Is there any quick points, or you save your points at the end? That's fine. Okay, we have eaten into your question time a little bit, but now Mark can give us an update on dormancy and sprouting.
Thank you. I'm Mark Taylor. I'm from the James Hutton Institute, also in Dundee. I'd like to thank the organisers for giving me the opportunity to talk about some of the work we've been doing on dormancy and sprouting in potatoes. As we heard yesterday, and I don't really have to introduce this to this audience, that potatoes are stored on an industrial scale. In the UK alone, there's about 4 million tonnes are stored per year. And in order to prevent premature sprouting, chemical inhibitors of sprouting are used, particularly CIPC. And we heard about some of the pressures on use of CIPC and the problems of using it. Also, cold temperature storage is used to prevent sprouting. This also has a high energy input and costs for the industry. So, I mean, despite the scale of this process, there's surprisingly little known about the dormancy properties of potato. We know different varieties have very different dormancy characteristics, but the basic biology of this process and bud release and sprouting are not well understood. And so we put together a project on controlling dormancy and sprouting in potato and onion, also in the BBSRC Horticulture and Potatoes initiative. We included onion because basically bud dormancy release processes and different storage organs are likely to have very similar biochemical and biological mechanisms, and we thought we could learn between the two crops. So the project's a collaboration between 4 academic groups, ourselves, University of Greenwich, Cranfield University, and Imperial College, and we're also, as well as getting support from the BBSRC, we're very grateful for support from the AHDB, PepsiCo, and Albert Bartlett's. And basically, we want to use a multidisciplinary approach. So within the academic groups, we have complementary skills. And it's a really exciting time in plant biology in general. I mean, we've heard about the advances brought about by the genome sequencing. Potato genome was published in 2011. That really has been a game changer for potato genetics. But also in fields such as biochemistry, molecular biology, there's been really great advances. So now we can measure metabolites at incredibly low levels, incredible accuracy. So we can measure plant growth regulators, and we can measure which genes are switched on or switched off using RNA sequencing. An additional feature of this project is that we have 2 associated studentships which will focus their development and plan for the future. So basically the genetics approach, which has really been the focus of what we've been doing at the James Hutton Institute. So we look at the offspring of a biparental population. So population contains about 180 to 200 genotypes, and essentially they all have the same parents, so they're all brothers and sisters. And what we can do is run gene markers on each of these genotypes, several thousand gene markers. We call these SNPs as the marker technology we've used. And if we measure the traits in these 180 genotypes, we can associate the markers with the trait. So some genotypes will have it and others won't. And when you do all that, you can see which markers are associated with a trait. And then we can, because we know exactly where the markers are in the potato genome sequence, we can identify the chromosomal regions that are controlling this trait. And the causative gene that underlies this region of the chromosome. We call it a quantitative trait locus, or a QTL. So variation of that causative gene are likely to be causing the trait. So we can look for the beneficial versions of this gene, which we call alleles. And then once we have markers for those alleles, they can be targeted a lot more accurately in breeding programs. But of course, the resolution that we have is still not perfect. So underlying these QTL regions, we could have 100 genes in here. So we need supplementary information about the biology of the process to help us decide which is actually the causative gene in this region. So here we can look at all the transcripts that are switched on in the bud as it goes through dormancy release. We can measure the levels of hormones or plant growth regulators during this process. And that will help us decide which is actually the causative gene. So this is the phenotyping data. So for each of these 180 genotypes, we've measured the sprouting characteristics of these tubers when stored at 10 degrees centigrade. And we can see that there's very different behaviors within the population. And the red and blue show the data from 2 seasons, which is actually quite reproducible. And we see this big variation. We can measure traits such as the rate of sprout growth, but also the time to dormancy break. And then we can use these data to try, in our quantitative trait locus analysis, to try and pinpoint the regions of the chromosomes that are controlling the dormancy-related traits. So in this way, we can see how many of these regions there are in the potato genome and the size of these effects. So this is what we call the genetic architecture of the trait. And we found 4 or 5 of these QTL that are consistent over the 2 seasons we've been looking at it. So this is just a bit more detail about the population we've used. We've used a diploid population. Most commercial varieties of potato are tetraploid, which makes the genetics a lot more difficult. But we developed this cross, which we can do the genetics quite effectively. These are the potato chromosomes, and this is the position of all the markers I was talking about, so we see where they all line up. And the cross contains some Solanum foreca, which is a wild potato species, which has very shallow dormancy, crossed with tuberosum. So there's a very wide variation of dormancy characteristics in this population, which is what you need for this sort of analysis. So for one of these regions, we can look to see which genes actually underlie the QTL, and there was a very obvious, we thought it was a reasonably obvious candidate gene to be controlling the trait. So if we see that, how do we proceed to prove that it is actually the causative gene? Well, here we make transgenic tester lines where we can switch the gene on or off and then see what effect it has on the trait. So these plots represent sprout growth curves from tubers in storage. And here we can see when we switch the gene off. In the red and blue, we have the control genotypes that are not changed. Here we have the lines where we switch the gene off. And the more we switch the gene off, the more rapid sprouting we get. Similarly, if we make transgenic lines where we turn the gene on, and look at the sprout growth curves, the control again in red and blue, and as we turn the gene on more and more, we see slower sprout growth. So this encourages that we have actually identified the causative gene. We can now start to look for naturally occurring versions of this gene and markers for those particular versions that we can use in breeding. So we're also supplementing this with transcriptomics. So we isolate buds at various stages during the dormancy release process. We isolate the messenger RNA, which is an indicator of which genes are switched on, and we can get a very detailed pattern of gene expression. And comparing fast sprouting and slow sprouting cultivars, we can gain some information about the biology of the processes that are going on. And in the same samples, we look at metabolite levels, things like the plant hormones and plant growth regulators. So this work is currently in progress. So to summarize, we want to understand the major genetic factors that are controlling dormancy and integrate those into breeding programs so we can tailor the variety to particular requirements, either fast sprouting or slow sprouting. We've identified some of the major genetic control points and the genetic architecture of the trait. And there's new breeding techniques that are being developed which will accelerate potato breeding in the coming years. Thank you. Oh, actually, sorry, I should acknowledge the people who have actually done the work in the various institutions. Glenn Bryan is leading the project at the James Hutton Institute. Leon Terry is a project leader from Cranfield. Colin Turnbull, and from the University of Greenwich, there's Richard Colgan and Debbie Rees, as well as the input of the industrial sponsors. Thank you. Finished now.
There's about 6 minutes left for questions. As I say, we've eaten into some of it. If you could please just shout out your name and where you're from ahead of any question. And for any of our 3 panel members who think have rushed through some fairly complicated research in a very short space of time. So have the first question.
Ryan Fenton, SRUC. It's a question about nematodes. You said you've done some plot tests for the genes, etc. Does anything get through the diffusing signal there to get added to the roots and successfully produce an infection?
Yeah, we've done more than plot tests. We've done the anti-feeding strategy has been tested in the field, in potatoes, in rice, and in bananas, and been shown in a number of field trials. The anti-feeding the anti-invasion defense the question is about has been tested in the field in potatoes, and it's also been tested in the field in Africa in bananas for subsistence farmers. Some animals do still infect the plant. The level of resistance that we see is about 80%, and we think probably the reason that you still get some animals infecting the plant is it's very, very difficult in the plant root system to get strong expression of a GM trait everywhere in a root system. So it might not be so strongly expressed in older parts of roots or places like that. But the thing I would say is that the world's going this way and legislation's going this way for GM, that you shouldn't just have one trait. You'd need 2 or even better have 3 traits to deliver what you want. So even if one of them goes down, then in terms of nematodes, you'd still have more than 90% resistance.
Our organisation resistance, you now get resistance in 3 different mechanisms, so they can adapt to even 3. But have you actually seen, is there a change in the nematodes whereby they might find this attractive as opposed to repellent?
No.
So populations could be adapted.
No. Populations, no, you're quite right. Populations could adapt. That's always a risk. But just for example, they can adapt as well as adapting to any GM-type defense. They can adapt to natural defenses. So, the potato industry is dependent on the H1 gene for resistance against raw stock, but we haven't seen any adaptation to that. The entire wine industry is the entire wine industry. They're all grafted onto pretty much the same rootstock that gives resistance to a mite, and that's controlled by one gene. And you think there the selective pressure would be enormous for that breaking, but around the world in all the vineyards, nobody's seen breaking of that thing. So what drives evolution of defense breaking? I don't know.
Yeah, I think the high-headed fox prefer
Does the root exudate effect also affect hatching?
No, the root exudate effect is specifically for attraction. It's not an anti-hatching effect.
So, do you get any effect on the promotion of hatching from the exudate?
No, we don't get any suppression. We don't get any enhancement of hatching from those plants. And we can just collect them in tissue culture and collect really concentrated liquid that you'd never get in the field, and we can't detect any effect on hatching.
Story. You're putting in 3 genes for blight resistance into the line. Kind of back across to David's work. We've seen with other blight GM plants that resistance can break down because the populations have evolved and changed.
Yeah.
So I'm just thinking forward, and the types of modelling that David is doing. Can we perhaps see ways that if you get 3 blight resistance genes in there, and we understand which genes are in there, and we understand which populations of blight are around, we can best manage spatially which fungicides that we use to control the blight, so we both prolong the life of the resistance genes in the plant and prevent the development of resistance to the fungicides. It passes both sides. So appreciate coming from David and how that could be used to potentially prolong the life of a GM.
Yeah, I'm not sure which genes specifically are going in, but I suppose the response is independent of those genes. You're right. I mean, some genes, even classically bred genes, have been shown to fail in the past. So the great thing about the GM, I think, is the intelligence that goes into the backstory behind selecting the appropriate genes in the first place, and an ability to chop and change those genes according to the pathogen population and the way they're deployed. So some modelling work that Pete Skelsey did actually as part of his PhD tested different patterns of deployment in single strips within a plant, within a field. Obviously not practical, but he was testing all the different levels of resolution. So testing blocks compared to strips, compared to blocks in different regions of the country. And certainly he proved on a theoretical basis you could slow down the selection process by choosing the way of growing those. Clearly what we want to do is avoid that, yoo-hoo, we've got a new blight-resistant potato, let's plant it everywhere, which would be fairly foolish. Might be very commercially successful for a few years, but long term that wouldn't be a good strategy. And balance that with, well, what is the correct approach? I'm certain we will still need to apply fungicides to suppress the lineages of blight which do overcome that. We want to be able to suppress those with fungicides. So we will still need a repertoire of active ingredients, a mixture of active ingredients on the fungicide front, combined with the good combination of genes. And what I tried to get over is that the knowledge of the underpinning information will help us in that. I don't know if there's a particular comment on the blight genes that have been used in this case.
No, it goes back to that first question about overcoming resistance. There is even one of the well-studied Blue 13 has now overcome one of the well-studied and characterized resistance genes. So the type of integrated pest management that the question suggests will be an absolute necessity to protect the GM trait for as long as we can before kind of like the next generation of GM traits for late blight becomes possible. Because I think having a GM trait for late blight that has longevity might be far more difficult than it is for nematodes. And I think it would have to be tret in the same way that, for example, Artemisia for malaria is tret. It must be deployed with great care in those areas where it's essential to deploy it, because the pathogen will quite likely at some point overcome it. So I think IPM is the way forward. There's no silver bullet to any of these things.
Okay, folks, I'm afraid that's all we've got time for. If you're staying for the next seminar, you can just stay where you are. If not, those of you, can you clear the room relatively quickly? Because we have eaten into the next time. Thank you.